Welcome to the digestive system lecture. This PowerPoint is going to run kind of like the kidneys PowerPoint, where I break it up into a few videos. So this is video number 1. So the digestive system is the process of you ingesting food, breaking it down, which is called digestion, and then absorbing the food into your bloodstream to provide your tissues with nutrients. Um, with this, we're going to start with some definitions. Just know that there are hidden slides from this presentation that you will have on your end that you are still responsible for. Most of those slides are anatomy slides where they should be a review, but if not, you can go ahead and go over them on your own. So we'll start with the definition of digestion. Digestion is the physical or chemical breakdown of food. You have mechanical digestion, which is physical pulverization of food. It's physically breaking it down using things like your teeth, like the muscular churning action of the stomach. And it does this in order to break the food down into smaller components in order to increase the surface area for your digestive enzymes to work on. Now, those digestive enzymes are part of a process called chemical digestion. And chemical digestion does what's called hydrolysis reactions. Hydrolysis reactions are types of reactions that break down macromolecules like multi-chain sugars or multi-chained amino acids into smaller monomeric subunits. And if you know this prefix mono, it means one. The whole point of doing both of these digestions in series is to break down the nutrients that you're eating into the individual components. For example, polysaccharides are broken down into monosaccharides, which are your sugars. Proteins are broken down into individual amino acids. Fats will be broken down into monoglycerides and fatty acids. And then nucleic acids are broken down into nucleotides. Only once you're in these forms are you able to then absorb that nutrients in the small intestine into your blood to be used as forms of nutrients. Now, we'll start from the mouth and work all the way down to rectum and anus for defecation. And we're going to start with you putting food into your mouth. You put the food into your mouth and you have saliva in the mouth. And saliva's function is to moisten the mouth, to begin starch and fat digestion. We'll talk a little bit about this in just a moment. Saliva cleanses your teeth by inhibiting bacterial growth. It's able to dissolve the food particles and chemicals so that they can be received and recepted by taste buds. And then it binds all of that food together, mixing it with the saliva to create something called a bolus. And a bolus is a key term that you should know. So bolus is going to be food and saliva mixed together. Now, saliva itself is mostly water, and it is considered hypotonic. It's made up of all these ingredients. You do need to know these ingredients. I'm going to go through each of them now and give you their functions. Salivary amylase and lingual lipase, these two are the enzymes found in your saliva. It is because of these that you're able to do starch and fat digestion in the mouth. It is very, very little starch and fat digestion, but nonetheless, you do begin breaking down starches and fats inside of your mouth because of saliva. Salivary amylase breaks down starches. Lingual lipase breaks down fats. These two enzymes will only work in the mouth because once you swallow food and saliva, they're eventually going to drop down into your stomach. And what do you know about the pH inside of your stomach? Well, the stomach is full of acid, so the pH is going to be really, really low. These enzymes that normally work in your mouth, they like to work at a pH of about 7.3, so pretty close to neutral. Once enzymes that like to work at neutral pHs go into extreme pHs like the ones in your stomach, these enzymes will get denatured and broken down and they will no longer work in your stomach. So these two enzymes are strictly in your mouth. Saliva is also made of mucus. Mucus is kind of like a goopy material and it's there to lubricate all of that food, lubricate the bolus that you're making, because it is this bolus that you're eventually going to swallow down your very small esophagus. And you definitely don't want the bolus getting stuck in the esophagus or having trouble getting down. The next ingredient is lysozymes. These are types of enzymes that kill bacteria. This is not an enzyme that breaks down food. It is simply there to help with cleansing of the teeth and inhibiting bacterial growth. The next one here is called an immunoglobulin A, IgA. There's a whole class of immunoglobulins, IgA, IgE, IgI. These guys are just antibodies. And antibodies are part of your immune system. These guys also participate in inhibiting bacterial growth and cleansing your teeth. So we're going to put an arrow to this one and to this one. The last one are electrolytes. Of course, like every other compartment and area of your body, you do have salts like sodium, potassium, chloride, phosphate, and bicarbonate. So this is your saliva. Now, salivary glands are found in your mouth, and there are 3 salivary glands you should know. You have the submandibular gland, which is underneath the mandible. You have the sublingual gland, which is underneath the tongue, and I believe if one got deleted, this right here is called the parotid gland. So Parotid. So 3 glands. Parotid gland. I'll number them for you. This is number 1. This is number 2, submandibular. And this is number 3. We're just going to number them to clarify. So the parotid gland, this one's found in front of the ear. It's on both sides. The sublingual gland is right under the tongue because sub means under, lingual means tongue. The submandibular gland is under the mandible because, again, sub means under, mandibular means mandible. Now this submandibular duct, I just want to point out, do you see this like white duct? This is a duct that goes from the submandibular gland through the sublingual gland and eventually into the mouth. You see a duct from the parotid gland as well into the mouth. These ducts are what allow the saliva that's created in the gland to actually make their way into your mouth. This is a picture of what you have in each of those glands. These guys are in the salivary glands, and they're made up of a few different types of cells that help to produce saliva. You have mucus cells, which are the tips right here. You can kind of see how bulky they are. And mucus cells, like the name states, make mucus. Then you have serous cells, the pink ones. The serous cells are what are going to make your enzymes. And then you have this whole branch here, which is made up of both mucus cells and serous cells. We call these guys mixed acinus. Each of these branches is called an acinus. You have a mucus acinus, you have a serous acinus, and then you have 2 of these mixed acinus. Again, the mucus ones make mucus, the serous ones make enzymes, and then the mixed ones make both. They secrete their fluids through the salivary duct, which will eventually open up into your mouth. Now, who controls the salivary glands? Well, your brain has a huge involvement with it. Inside of your brain, you have a region called the medulla oblongata and the pons. Hopefully you remember this from anatomy. These areas have regions inside of them called the salivary nuclei. Nuclei is just a region where you have clusters of neurons. The salivary nuclei is responsible for generating the signal to create saliva, usually in the presence of food. So the salivary nuclei receives its signal from your senses. You smell food, you see food, and you think about food. All 3 of these are going to stimulate the salivary nuclei to make saliva. It's just like if you were sitting here and I gave you a Sour Patch Kid in your hand. And I said, don't eat it, just look at it. and get it really close to your mouth as if you were going to eat it. Just having that Sour Patch Kid in your hand, making you think about that sour flavor in your mouth, it's going to cause your salivary nuclei to start producing saliva. So with that, because there's a nervous system involvement, there's going to be, again, two modes of control: the parasympathetic stimulation and the sympathetic stimulation. The parasympathetic stimulation, the rest and digest one, well, the word digest is in there. If you're resting and digesting, you sure as heck better want more saliva to help you coat the food. The parasympathetic stimulation is going to increase production of saliva. Then you have the sympathetic stimulation, and this is going to decrease production of saliva. So if you ever notice when you're really scared, really excited, or working out really hard, those are all ways to stimulate your sympathetic nervous system. And in all three of those situations, you notice that your mouth is not only dry, but your saliva is very, very thick. A lot more mucus, a lot less water in there. And that's because you're down-regulating your salivary nuclei. So let's say you've put the food in your mouth. Your salivary glands have produced the saliva. The saliva has mixed with the food to create what's called the bolus. Again, that key term is right over here, you guys. At this point, you're going to go through the process of getting this green-colored bolus, just for illustration purposes, not green in real life, unless you've eaten green peas or something, to push that bolus to the back of your throat using the muscles in your tongue. From there, the bolus goes down and it goes through what's called the esophagus. The opening in front of the esophagus, a little bit larger, is going to be your trachea. What's supposed to go down your trachea? Air. And what is supposed to block that bolus from going into the trachea and blocking it and you choking on the food? Well, you see this piece of cartilage here? That's your epiglottis. Notice how it closes to direct the bolus to go into the esophagus. Now, once it's in the esophagus, do you sit there and physically control the muscles in the esophagus to get the food down? You're like, all right, go, push, 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 all the way down? Well, no. Once it's in the esophagus, you don't have control over the wave-like motions that actually push that bolus down. Those wave-like motions are called peristalsis. It is in red and it is in bold. Please make sure you know this word. Peristalsis is smooth muscle contractions that are involuntary that work in a wave-like fashion. You see this wave-like fashion? You got one bolus here, you got another bolus there. Those smooth muscle contractions called peristalsis are not only going to happen in your esophagus. We're going to see this come up again several times. So be sure to remember the definition of this term. Wave-like contractions that help push things through the digestive system. Now, this is the step-by-step for how this all happens. What I want you to underline on this is upper esophageal sphincter and then lower esophageal sphincter. So the upper esophageal sphincter is going to just be the sphincter that opens and closes right here at the top of the esophagus, and we'll put a little arrow next to that. And once that bolus gets going down, at the end of the esophagus, you're going to have what's called a lower esophageal sphincter. But, surprise, surprise, there's two other names for this that you're going to see soon. Another name for the lower esophageal is the cardiac sphincter. Another name for it is the gastroesophageal sphincter. It's simply the sphincter that opens and closes to allow the bolus to go from the esophagus and plop into the stomach. Now, why do you think it would be important to have a sphincter between the esophagus and the stomach? Think about what's in the stomach. Well, there's a lot of acid in the stomach. What do you think would happen if acid jumped out and went into your esophagus? What does that feel like for you as a human? If you said heartburn, you're correct. If you don't have control over that sphincter here and it's constantly open or palpitating, acid from the stomach can jump back up into the esophagus, and that feels very painful to some people, and it's called heartburn or acid reflux. So now that we're at the stomach, we're going to kind of transition into first the structures that are important, and then we'll talk a little bit about the physiology of it. So we went from the esophagus through that lower esophageal sphincter, also termed the cardiac sphincter, and now we're going to get that bolus to drop into the stomach. The stomach itself is just really a storage facility. Its primary function is to store food, although it does have other functions of mechanical and chemical digestion. The stomach's able to do mechanical digestion by churning the food and crushing it using the smooth muscles that are found covering the stomach. If you remember from anatomy, the stomach is covered with 3 layers of smooth muscle, all going in different directions in order to be able to churn in different directions. The stomach also does chemical digestion of food, specifically proteins and fats. So if you recall, in the mouth, you started digestion of starch and fat, no proteins. Now in the stomach, you're doing a little bit of chemical digestion of proteins and fats, no starches here. Now here's another key term you gotta know, and that term is chyme. This is gonna be a mixture of bolus plus acid. So it's a soupy mixture, almost pasty, of the semi-digested food that came from your mouth that dropped into your stomach mixed with acid. That is what chyme is. And we're going to keep this term until we get through to the large intestine, kind of colon area. Now, most digestion will happen after this chyme goes into the small intestine. So although we did start breakdown, aka digestion, of starches, proteins, and fats in the mouth and the stomach, you're going to actually do most digestion in the small intestine. So just as a brief review, the stomach has a few different sections. We've got the opening, which is the cardia region. You've got the fundus, which is this top dome. The majority of the stomach, which is the body, and then the end of the stomach, which is the pylorus, which leads through the pyloric sphincter into the first section of the small intestine called the duodenum. Now, this is where we get into a little bit of the physiology. There's not much mechanism here, but it is about the innervation and circulation of the stomach. So just to introduce you to this illustration on the right side, you have a stomach here. You got the pancreas down below, you've got a spleen, the liver, the small intestine, and then the large intestine. And what you notice is that you have veins attached to all of them, and they take blood from the small intestine, large intestine, nutrients from the stomach, pancreas, and spleen, and they bring it all into the liver for filtration purposes. So with this, the vascularization allows us to take things like waste or nutrients or drugs or alcohol through the venous system and put it into the liver for filtration and cleanup. This whole system of veins that goes to the liver and then eventually to the inferior vena cava is called the hepatic portal circulation. Now, the hepatic portal circulation has the term hepatic in there, and hepatic should always refer to liver. So this hepatic portal circulation is going to filter everything that we collect from the digestive system before returning that blood back to the heart. Now, in terms of innervation, this illustration doesn't show you very much innervation in it, but the innervation of the stomach can be both parasympathetic and sympathetic. And if you recall, what we used to call the parasympathetic nervous system was the rest and digest nervous systems. So In the parasympathetic nervous response, you're definitely going to be upregulating the stomach using a nerve called the vagus nerve. Make sure you know that nerve name. Now, on the other hand, you do have sympathetic innervation as well, which is going to downregulate the stomach, and that's through what's called the celiac ganglia. Now, biggest difference between these two nerves is that the vagus nerve is a cranial nerve coming straight from the brain, and the celiac ganglia is a cluster of neurons that comes from the spinal cord. So again, parasympathetic upregulates the stomach to churn more, make more acid, and the sympathetic nervous system is going to downregulate the stomach to decrease the muscle contractions and decrease production of the gastric juices. Now, those gastric juices, the acid, all of the fluid in the stomach is made from the glands of the stomach. Now, if you remember, these ridges in the stomach are called rugae, but what you might not have known is that in those ridges, you have pits that go all the way down. And if you notice, these pits, they're lined with cells, different colors to indicate different types of cells. The opening is called the gastric pit, and the bottom tips are called the gastric glands because it is these glands that make secretions that are eventually going to go out into the stomach. Secretions like acid. Other secretions that include digestive enzymes. So these secretions are made of mostly water, the acid, and then the enzyme pepsin. We're going to talk about this in depth today. So let's take a look at two types of common glands in the stomach. In the stomach, you've got the pyloric gland and the gastric gland. And one of the biggest differences you'll see is the density of cells in those glands. In the pyloric gland, you've got a lot of these mucus cells, which you now know make mucus. You also have parietal cells in orange, and you've got G cells. In the gastric gland, you have, again, mucus cells, you've got parietal cells, and you have G cells. But what you have in this gastric gland that you don't in the pyloric gland is a cell called a chief cell. Now, the objective here is to understand the functions of the glands based on the types of cells they have. So here are three of the main cells, and I'm going to give you their functions. You should probably star this slide because this is a very, very important slide in terms of test questions on your next exam. So the first one, mucus cell, pretty easy one, made by the mucus cells to produce mucus, and that mucus is there to act as sort of a coated membrane inside the lining of the stomach. We'll talk a little bit more about that later. The parietal cells, these guys here, they kind of look like the letter W. The parietal cells, their big job is to make hydrochloric acid. This is the acid that's found in your stomach. It's the typical stomach acid. But that's not its only job. Parietal cells can also make a hormone, which we call ghrelin. Ghrelin, right over here, is a hormone name you should know. And ghrelin is produced by the parietal cells to signal your brain that you're hungry. The more ghrelin you make, the more hunger you feel. The last cell here is a chief cell. And the biggest job of chief cells is to make your digestive enzymes for the stomach. Now, these digestive enzymes will only work in the stomach because remember, enzymes only like to work at specific pHs. If you're making enzymes that only work in the stomach, what pH do you think those enzymes like to work at? Well, probably really acidic pHs. So the chief cells will make two main digestive enzymes. The first one is called gastric lipase. If you recall when we were talking about the mouth, there was an enzyme that I mentioned called lingual lipase. Lingual refers to mouth or tongue, and lipase means enzyme that breaks down lipids. Gastric lipase, gastric or gaster, this always means stomach. So this is a type of lipase that is found in the stomach. This breaks down fats in the stomach. The second enzyme is called pepsinogen. It ends in ogen, which means it's inactive. Any enzyme that I ever give you that ends in the suffix ogen means that it is inactive. So pepsinogen is the inactive version of pepsin. I will teach you how to activate it, but just for now, know that the chief cells make the active lipase enzyme and an inactive version of pepsinogen. So here's your first big mechanism for digestive system. Be sure that you know this mechanism front and back. So we will start with getting me a pen. And we're going to start with step 1 right over here. So step 1. You know this reaction already, which is really great because you don't have to memorize another one. But this reaction, surprisingly, is the same reaction that's found in the respiratory system for carbon dioxide dissolving in plasma. So you take carbon dioxide from the blood, you move it into your parietal cell. In the parietal cell is where you have that typical cell reaction that you learned about in respiratory system. CO2 plus H2O, using an enzyme called carbonic anhydrase, is going to turn into carbonic acid. Carbonic acid spontaneously dissociates into proton and bicarbonate. This bicarbonate, we really need it to go back into the blood because what do we know bicarbonate acts as in the blood? It acts as a buffer. But what we really needed here was the proton. More protons you have, the more acidic you become. And the whole goal of the stomach acid is to be very acidic. So this proton will then use this antiporter, the proton potassium antiporter. You're going to take a potassium from the stomach and move it into the parietal cell against the gradient. Then you're going to take a proton and move it out into the stomach acid against its gradient as well. Because they're both moving against their gradients, you're going to require ATP to do this. So it's called the potassium proton ATPase. So you've now successfully put the proton into the lumen of the stomach. Now, the type of acid that's in your stomach is hydrochloric acid. So HCl. So we have the H here. Now we got to take the Cl from the blood and transition it into the lumen of the stomach. This chloride will move openly and freely. We don't need to get into the mechanism of it, but it will require exchange of other negatively charged ions between the cells. But the gist of it is that you're able to move the chloride from the blood into the lumen of the stomach to combine with the proton and make HCl. So be sure you know the mechanism of hydrochloric acid formation in the parietal cell. Now, why do we need stomach acid? What's the point of it? Stomach acid, let me just make very clear, is not an enzyme. Stomach acid does not digest nutrients. Stomach acid's number one function is to activate pepsinogen into pepsin. You will learn the mechanism of this in just a moment, but that is point number 1 over here, to activate pepsin. Stomach acid does have the ability to break up some plant tissues. It has the ability to convert ferric ion to ferrous ion. Really what you see here is that ferric ion has a 3+ charge, ferrous ion has a 2+ charge. Might not seem like a big difference to you, but by just adding an electron to it and becoming ferrous ion, this is a huge benefit for you. Why? Well, us as humans, we can't use ferric ion. Ferric ion is really no benefit to us in terms of the type of iron it is. But ferrous ion, this is the type of iron that's going to be found in your blood. So your blood tastes like metal sometimes whenever you have a chance to taste it because it has iron in it. So ferrous ion is required to produce properly functioning hemoglobin, and it is in that hemoglobin that you find the iron. Lastly, acids have a huge ability to kill bacteria, so it acts as a pretty good immune response to anything pathogenic that you might have ingested. Now, here's that mechanism of activation of pepsinogen. I'm going to go through it pretty slow, and I'm going to kind of tie it all together for you. Here's that gastric gland. We've got mucus cells, parietal cells, chief cells, you got the dark pink G cells, but for this mechanism, we're only going to focus on parietal cells and chief cells. So up till now, you've learned what each of the cells makes. You know that parietal cells make HCl. You know that the chief cells make pepsinogen, inactive version of the enzyme. How do you activate pepsinogen? Well, hydrochloric acid has a cleaving effect. The word cleaving means cutting. HCl has the ability to break this bond right here between these two amino acids. Once you break this bond here and remove a portion of the peptide, you now have the activated version. So you take pepsinogen, cleave off the inactive part, and it turns into the active form of pepsin. Pepsin is now the active enzyme, and go ahead and write this down on your notes. Pepsin's job is to digest proteins. So it can take long chains of proteins and break it up into smaller, partially digested chains of proteins. Now, what's really cool about this is that once you make pepsin, pepsin can start to work like HCl in the sense that it can go back and cleave more pepsinogen to activate it into pepsin. This is called an autocatalytic effect. So not only is HCl working to activate pepsinogen into pepsin, but the pepsin that you produced also can go back and activate more pepsinogen into pepsin. So the acid in the stomach is required to activate pepsinogen into pepsin, and pepsin begins protein digestion. Now again, if you recall in your mouth, the only two enzymes you make are amylase and lipase. So in your mouth, the only two things you can start breaking down are starches and fats. Only once you get into the stomach can you start then breaking down proteins and fats. Remember, in the stomach, you make lipase and pepsin, the enzymes that break down fats and proteins. There's no enzyme in the stomach that actually breaks down starches, so you will halt starch digestion temporarily once you're in the stomach. So this is where we're going to pause. I will pick up on this slide in the second part of this video.